Hello everyone. I would firstly like to thank the organizers for providing me an opportunity to talk at the Dutch Biophysics 2021 conference. My name is Mitra and I'm a postdoc at the laboratory of Professor Erwin Petterman at the Vrije Universiteit Amsterdam. In this talk, I am sharing some of our new research findings on understanding how ciliary proteins assemble into trains at the cilia base and enter the sensory cilia of C. elegans. Right. Before I get ahead of myself, let's have a quick introduction. Numerous cell types in our body have protrusions that extend out of the cell membrane, called the primary or sensory cilia. These cilia can sense extra changes in the external environment and communicate it back to the cell, thereby acting as signaling hubs of the cell. When we peer into the cilia, we have a highly intricate transport machinery called intraflagellar transport that is responsible for the maintenance and functioning of the structure. There is a microtubule-based axoneme that provides the template for ciliary motor proteins to drive intraflagellar trains back and forth throughout the length of the cilia, bringing in new material into the cilia and cycling out waste products. In this study, we are essentially asking how the ciliary proteins reach the cilia base, assemble into trains, and cross the diffusion barrier to enter the cilia. To further dig into this work, it is crucial to know the essential components of anterograde IFT trains that enter the cilia. There are kinesin motors, two of them in the system that we study, that drive anterograde transport from the cilia base to the tip. Dynein motors, responsible for retrograde transport, are present as cargo. The IFT trains are essentially complexes of several proteins that can be divided into two core subcomplexes called IFT-A and IFT-B. Further, there are several cargo proteins, including ciliary membrane proteins and channel proteins like OCR2 that dynamically associate with the trains. The model system that we use to study sensory cilia are millimeter-sized worms called C. elegans. These organisms are easy to maintain, transparent, making it possible to image inside their body, and very well characterized due to decades of research. They have about 1,000 cells, 302 of which are neurons, 60 of which are ciliated. We image four such sensory neurons called the PHA and PHB neurons, which are located near the tail of the worm, with the neurons arranged into two pairs. The geometry of the sensory neurons is interesting. They have 8 micrometer long cilia that are separated from the cell soma by approximately 100 micrometer long dendrites. This further raises an interesting question on how ciliary proteins that are being expressed in the cell soma reaches the cilia base. Right. To observe transport inside the pair of cilia and more generally in neurons, we generate worm strains where one of the ciliary components is labeled with a fluorophore. Here you can see standard wide field fluorescence imaging of the cilia of a worm where one of the IFT train complex proteins is being labeled with GFP. You can very nicely see the transport dynamics with IFT trains moving back and forth in the two cilia. We perform two slightly sophisticated optical tricks in this work. Firstly, we can do single molecule imaging where we essentially photo bleach the fluorescent proteins using high power laser excitation such that only a few protein molecules fluoresce at any given point, which can then be visualized. For instance, here we see the motion of single ciliary dynein motors inside a pair of cilia. We can track these molecules with nanometer precision, thereby gathering subtle insights into the dynamics of the individual proteins. The other trick we use involves manipulating illumination beam size. When we normally perform wide field imaging, the illumination beam size is rather big. However, our region of relevance, that is the cilium or the section of the dendrite, is only about 10 micrometers. So if we illuminate only the relevant region by reducing the beam diameter, we only bleach the fluorescent proteins in that area. Any new molecules that enter the region can then be readily visualized because they are not being bleached. Okay, we start off by imaging ciliary components in the dendrite. Firstly, let's have a look at ciliary dynein. As you can observe, the dynein motors undergo rapid diffusion in the dendrites. We can track individual dynein motors and estimate diffusion coefficients. And for dynein, it is about 0.7 micrometers square per second. When we do a similar kind of imaging and analysis for kinesin 2 motors, we find that the diffusion coefficient of these guys is 3 times higher than dynein, which is to be expected since kinesin motors are much smaller in size. We can similarly extract the diffusion coefficient for IFTB proteins like OSM6. And OSM6 is a much smaller molecule than even kinesin 2. Surprisingly though, we find that the diffusion coefficient of OSM6 is 2 times lower than dynein, which would tend to suggest that they are already present in the complex of IFTB, which is a much larger particle then. Similarly, we can extract the diffusion coefficient for membrane proteins like OCR2, which in the dendrite are presumably present in vesicles. And of course, these vesicles are then diffusing at a much slower, uh, with a much slower diffusion coefficient. So in summary, we find that the ciliary motors, which diffuse quite rapidly, for them, diffusion is sufficient to transport molecules from the cell soma to the cilia base. The slowly diffusing ciliary components, on the other hand, have devised a slightly different strategy to reach the cilia base. So when we start looking at IFT proteins like IFTB protein OSM6 in the dendrite, we very quickly observe that they are being transported in packets in a directed manner along the dendritic microtubules towards the cilia base. The velocity of these packets are between 1.5 to 2 micrometers per second. The variability in the intensity of different packets would suggest that different amount of material is being transported in different packets. Also, the time lag between consecutive packets that are being released from the cell soma seems to be exponentially distributed, which we can fit and get a certain characteristic rate at which packets are being transported to the cilia base. Interestingly, for the different proteins, the IFTB proteins, IFTA proteins, as well as ciliary membrane proteins, for all of them, we find a slightly different rate or a significantly different rate at which packets are being transported to the cilia base, which would suggest that they're all being regulated differently. The amount of material that reaches the cilia base is controlled differently for each of the different components. Next, we image ciliary proteins at the cilia base. First, let's have a look again at IFTB protein OSM6. In the maximum projection, you can see where the dendrites and the cilia base are and where the cilia is supposed to be. When I play the movie, you can see that the dendritic packets are moving towards the cilia base, very often pausing 0.5 to 1 micrometers before the cilia base, where most of the dendritic microtubules end. The second location where they pause is right at the cilia base, where they are expected to dock uh, uh, and repackage into IFT trains. Every now and then, you can also observe OSM6 molecules, the one that do not bleach, uh, also enter into the cilia. Tracking of these packets reveal rich pausing dynamics, as well as reveal the spatial architecture of microtubules at the dendritic endings. The pausing and the entry at the cilia base we can characterize a bit further. For events where we see entry into the cilia, we record a pause time and the location of the pause with respect to the cilia. The pause time for OSM6 is around 1.6 seconds, and the pause trains are located right at the cilia base. Next, we look at the ciliary motors, starting with the retrograde motor ciliary dynein, which acts, if you remember, acts as a passive cargo in anterograde trains. As you can see, they are diffusively present in the dendrites. Every now and then, a dynein molecule would dock onto the cilia base, presumably to the train being assembled. For some of them, the ones that do not get bleached, you can also see them enter the cilia. When we characterize the pause times similar to what we did for OSM6, we find that the pause time is slightly lower. The pause location, however, is still right at the cilia base. When we look at the anterograde kinesin motors like OSM3, the story is slightly different. Again, they are diffusively present at the dendrite. At the cilia base, however, it appears that they catch on to already pre-assembled trains and directly enter without pausing at the base. Indeed, when we characterize the pause time, we find that it is much lower, 0.3 seconds in case of OSM3. Also, the starting location where they dock onto trains, it's not just at the cilia base, but also deeper into the cilia, suggesting that these motors can diffusively enter the cilia and attach to already moving trains. So to summarize, we find that ciliary motors are transported diffusively from the cell soma to the cilia base. In contrast, IFT train proteins as well as ciliary membrane proteins are transported in a directed manner in packets. These packets are carried till the end of dendritic microtubules, where they pause before docking onto the cilia base. At the cilia base, anterograde IFT trains are assembled with different components incorporating in a sequential manner. Once the IFT trains are ready to enter, they are picked up by anterograde kinesin 2 motors and they can enter the cilia. Thus, in this study, we have been able to directly visualize how different ciliary components reach the cilia base, package into trains, and enter the cilia of C. elegans sensory neurons. Finally, I would like to thank Professor Erwin Pettermann for providing me with such an exciting project to work on in such a delightful work environment. I would like to thank the different members of the WORM team, as well as the members of the Physics of Living Systems section in the Vrije Universiteit Amsterdam, for all the assistance and the fruitful discussions. And finally, thank you for your attention.